a PhD in neuroimaging application to neuroscience in the University of Havana. After that, he moved to Montreal, where he completed a first postdoctoral term at McGill University. And his work was so well recognized at that time that McGill offered him immediately a position, a tenure-track position as assistant professor, and been promoted to associate professor. He's a member of the Lumen Center of Neuroinformatics and Mental Health at McGill University Research Center for Age Studies in Aging, and he's also at the McConnell Brain Imaging Center. He is associate professor, as I mentioned, at the Montreal Neurological Institute, and his work is about obtaining as much information from data about mechanisms that are happening intrinsically in the brain associated with disease. So I know him a long time ago, but the first work that really got my attention was publication of this group in Nature Communication in 2016. So at that time, I was very much focused on how vascular dysfunction happen in some diseases related to that have some kind of neuroinflammation ongoing in the background. So like Parkinson, Alzheimer, TBI, they have normally, they have a common factor normally, which is neuroinflammation going in the background. And that neuroinflammation may be triggering some dysfunction of the vasculature. So his work in Nature Communication at that time was proving that the first thing that happened in Alzheimer's disease was the vascular dysregulation and the disruption of the brain-blood barrier. And at that time, I was much working on this, so it was a very, very stimulating work. And with that, I wanted you to join me in working with the
Hi everyone, it's a pleasure to be here, and for that I would like to thank the organizers, and in particular to Jorge here. I have to say that when I started in the Cuban Neuroscience Center about 20 years ago, over 20 years ago, Jorge's example and work was a kind of strong inspiration for me and my generation from that time, so for that, I really appreciate that. Today we will be talking about the work that we have been doing for characterizing neurodegenerative disorders from a multi-scale, multifactorial perspective, and how we can use these computational models or associated computational models to identify personalized therapeutic needs. Essentially, our lab at the Montreal Neurological Institute follows an integrative engineering perspective in which we try to combine as much data as possible or available for each participant or patient. We use multiple neuroimaging, including structural, PET molecular imaging, functional, vascular, night vision MRI for inference brain connectivity, and different other different modalities. We also use multi-omics molecular information, which will include genomic, epigenetic, transcriptomic, and the single cell level proteomics, metabolomics, lipidomics, which we also try to combine with environmental information, lifestyle information, socioeconomic factors, social interaction information, clinical and therapeutic information. And we believe that would allow us to create integrated computational models that would allow better not only to understand how the different biological factors interact at the individual level, but also how we need to, or what specific factors we need to target and in which specific way to conduce the person from a given pathological state to a decided condition, which could be very clinical symptoms or at least a point in the person declining in an accelerated way. However, with all these data modalities that I'm mentioning and others that exist, including EEG, MEG, etc., the number of potential markers, biological markers that we are talking about could pass 1 million. Iturria-Medina. And this implies like a classic way that we see in the field, in the neuroscience field, is like to go one by one, trying to correlate each of these markers, like I know how they quickly correlate with amyloid and tau, etc. And for us, that doesn't really work because we don't believe that actually we're going to be able to find one specific biomarker that will be able to reflect all the specific aspects of a given disease. And also the diseases that we are talking about, neurodegenerative diseases, they are highly heterogeneous, and in fact, no single feature should be able or will be able to capture the heterogeneity of the disease. And for that, this is exactly what computational disease progression model focus on. It's like they try to integrate data coming from different sources in order to understand or characterize individual disease progression and heterogeneity. And there are also other outputs that are about which I will talk later. Essentially, there are two main families of disease progression models, or computational disease progression models. One is the empirical models that I'm sure you are familiarized with. These are models like classic machine learning models, regression models, like any model that focus on making predictions but not necessarily giving you explanations on how the biological factors are interacting with each other. So, and not even a model that we could still open, like if we do a classic regression, statistical regression, yes, we could understand what specific factors have a stronger way compared with others, but we are not assuming or inferring there any causality between them. So those models still will be classified as empirical. On the other hand, we have the mechanistic brain models, which focus on understanding how the different biological factors interact, and they could or not offer predictions, but the main question is related to how exactly the disease is focused on the disease mechanism. I will present today some examples of these two major families and how we have used them and combined them to target some specific questions associated with neurodegeneration. One of the major challenges that we have when we study Alzheimer's disease or Parkinson, any major neurodegenerative disorder, is that they take decades to develop. They could take decades to develop. And usually, most of the knowledge that we have comes from group comparison. So when the person, let's say you have a control group that doesn't have the disease, and then you have the group that has the disease diagnosed already. However, by the time that someone is diagnosed with the disease or have the clinical symptoms, that person has been developing it for 20, 30 years. Which means that there are a lot of intermediary processes there that we cannot capture just by comparing them, doing a statistical analysis at the end. At the end, a lot of things are already altered, and we cannot really understand which were the specific steps or stage that conducive to that specific condition. So, another minor limitation is that most of the data that we have is not collected in a way that would allow us to understand what happens across all that period. And obviously, that happens for multiple reasons. One of them is that we cannot follow forward. We could follow a person for 30, 40 years, but the technology to get the best data or the data that we currently have only exists, like 10 years ago, 20 years ago maximum. There is not really any study so far that try to, there are some studies trying to capture the whole period, but the data is still not there. Well, about 5 or 6 years ago, we tried to infer the longitudinal or long time scale of Alzheimer's disease, but using cross-sectional data, but in a way that we could reorder the individual. Say you have a high enough population, this an heterogeneous population, you should be able to reorder the persons in a way that allow you to see which were the specific stages that by which the person went passing. Right, so you put first the people that is less affected, and then those that are a little more affected, and more affected, more affected. Like at the end, we should be able to think like this with trajectories or something equivalent like to a movie, which if we look at it, then it should give us information or longitudinal information of the whole process, and not only what happens when we compare the extreme or the most advanced individuals versus the no advanced at all individuals. So that's what we did, and from that, initially we obtained gene expression data from both brain from the brain or from blood from two different cohorts. Specifically from the Rosmar of Religious Order Study and Memory and Aging Project that is led by the Chicago University, or ANI, the Alzheimer's Disease Neuroimaging Initiative, maybe you know about it. And what we have here in the center is just a typical gene expression matrix. On the rows you have the number of subjects, on the columns you have specific genes or transcripts that represent specific genes that could go up to 40,000. So, and that's a lot of information. So it's a big matrix, but to analyze it, then we apply a specific constructive machine learning techniques that allow to identify principal components, but while maximizing or somehow giving more weight to those patterns that are present on the disease population and not on the background population. So this constructed PCA, there are other variants of this constructive dimensionality resolution techniques essentially give you or identify the enrichment patterns in the population of interest. So we apply that. We finish with some component for the whole population, like let's say 5, 6, and then we assume that the individual values on these components will allow you to represent in a multidimensional space each person in a way that the position of the closer the person will be to the center or to the origin will be the person will be closer to don't have any to don't have a cognitive impairment or to be like normal, cognitively normal individual. And the farther the person is from the origin, the more advanced will be the person in terms of developing Alzheimer's disease dementia. Then we use a specific methods to identify trajectories in that space in a way that those individuals, like, we just join in the brains after, right? So you go from those brains that are closer to the origin of not having the disease or not having symptoms, and then you back to the closer brain on that space and move to the other one or the other one, in a way that you identify distinctive sub-trajectories in that way, or distinctive ways of progressing to Alzheimer's disease dementia. Then we define it a given, we define the distance or the geodesic distance to the origin, which allows us to obtain something that we call the disease or gene expression disease progression score, or a gene expression pseudotime. It's just a number that gives you an idea of how advanced, how early or advanced is someone in the time scale of developing Alzheimer's disease dementia. The lower is the pseudotime, the better, or let's say the closer to 0, the person will be close to be cognitively normal. The closer is to 1, the closer will be the person, to develop Alzheimer's disease dementia. Then we verify that this gene expression pseudotime predicts without any training a level of neuropathologies, alterations of severity in the brain. Also predicts cognition and cognitive decline. Neuropathology was in both cases of the in vivo cohort and the postmortem cohort, and the cognition was also, and we verified it was predicted in both cases without being trained for that. But also that we could see cognitive decline, or we could infer cognitive decline in the person that was still living from just a blood sample. This is not a mechanistic model. We have here, but we still have weights associated with the specific genes, so we can go back and identify what specific molecular pathways, cell types are the most informative in terms of informing about the progression of Alzheimer's disease dementia. However, gene expression represents only of the multiple biological levels of factors that are involved in developing a given disease. We, in addition, say the genes, we first have the genome, and then these genes can be expressed or no, which depends on also multiple reasons, but one of them is on the epigenetic effects. So we extended this work to also include epigenetic information on the epigenome of each subject, the transcriptome, the proteome, and the metabolome of each subject. So now we extended the computational model to include, again, we use the constructive machine learning technique in a way now that we fuse the information coming from all these layers. Here we have, I don't know, over 200,000 markers, or easily over 100,000. And then we fused all this information using machine learning and extended the model to now identify distinctive sub-trajectories in that space. So what distinctive here means is that, for example, you can see AD sub trajectory 1, AD sub trajectory 2. In this case, the level of alterations, of molecular alterations that one sub trajectory is having are different to the level of molecular sub trajectory that the other sub level of molecular sub trajectory that the other sub trajectory is having. In a way that all these individuals might finish with the same clinical symptoms, but they are progressing or they have different biological mechanisms to get there. And this is very important because as we're going to see later, and this might explain, or this to some extent might catch why some people respond to a given treatment and others don't respond to it, which is one of the major issues that we have now with generalized medicine. Then again, we define it, so the main output of this model are now two scores or two values. Before, with the only gene expression model, we were getting a gene expression pseudo time. Now we get a molecular or multi-omics molecular pseudo time. Again, a value close to 0, give a proximity to normality, a value close to 1, and proximity to late AD dementia. But we also get subtypes here. So a person is now assigned to one sub-trajectory or another. And depending to what sub-trajectory the person is on, it means that the person is developing or following or being affected by very specific or the specific mechanisms affecting that disease variant. We verified that, again, that the pseudo time was capturing level of the neuropathology severity grade. Also that the three subtypes of sub-trajectory, the individuals from these three subtypes have different patterns of cognitive alterations in the brain. Sorry, of cognitive alterations, cognition is one of the outputs of the brain. And also that when we analyze the single cell data, or we identify what specific genes, proteins, metabolites were associated with each subtype, and we map that to a cell type alteration, single cell type alteration in the brain. We observed that each subtype was associated with a specific cell type alteration, which also somehow supported the idea that they were representing or they were capturing different disease mechanisms. Recently, we extended this work, and now this is under consideration. We started with only one molecular omic. After we included 4 omics, and now we have 8 omics in brain or measured in brain. And we extended the same model. So, well, essentially, we use again epigenomics. Now we have single cell transcriptomics and also bulk transcriptomics. We have proteomics, glycation proteomics, glycosylation proteomics, metabolomics, lipidomics. We fuse all that information. We identify different sub-trajectories or distinctive sub-trajectories, each of them having distinctive alterations across all these layers. And now we extend this work, well, in addition to including more molecular information, the second way in which we're extending this model is that now we try to understand, once that we identify the distinctive sub-trajectory, we do neuroregulatory analysis, or we perform neuroregulatory analysis for each of them, trying to understand how the cells interact and each of them, or for each of them, and what are the specific genes alterations and genes and cell type alterations that are characterizing each specific subtype. So essentially, we are passing from a more empirical model to doing some mechanistic analysis. So in a way that normally most of the knowledge that we have about how genes interact in Alzheimer's disease is coming from the whole population. So this allows us to go specifically to unique disease sub-trajectories. That should then allow us not only to understand better the disease, but also what specific genes and subtypes we need to focus to target better each disease variable. Then the third part of the new study, of the new approach, is that all these sub-trajectories analysis of this taxonomy for Alzheimer's disease dementia is performed in this case or in the previous cases with post-mortem brain data. And the reason is that normally we cannot get all these data modalities for or persons that are living because that's highly invasive, as you know. So normally what we do, we have access to this huge brain bank, which have been characterized in many different ways. So we are analyzing or trying to create a taxonomy of a very heterogeneous disease like Alzheimer's dementia using this post-mortem data. But if once that we identify at what stage of the disease the person or what subtype is following, we cannot use that in reality because that person is already there. We cannot do anything for that person. So the third step in this new work is that now we are training algorithms to to translate all the say information or all the taxonomy, subtypes, the drivers of the disease, etc., that we are finding in the postmortem data to in vivo or to data that was collected when this person was living. So fortunately, many of these individuals, over 1,000 of them, also were characterized when they were living. So we, or the British University, collected brain imaging from them, routinary blood tests, metabolomic, proteomics from blood. Also, they were characterized psychologically, they were characterized in terms of psychiatric symptoms, and they were characterized in multiple dimensions. They also have a RNA or gene expression quantification from blood, cytokines, cell-free DNA. And when we are doing that, for the matched individuals, for those that we have already a post-mortem brain characterization, that also have in vivo data collected, then we are training machine learning models to pass the post-mortem classification, the subtype and the pseudotype for the in vivo, for the time that they were still living. If this is successful, then we will have models that would allow you to pass information extracted when the person was dead to in vivo individuals, and then that can be used in the clinic later. So that can have a translation application. First, we identified again, there are many outputs from this new study, but from the most important results, we identified again 3 main distinctive subtypes. And when we characterize or try to identify the putative drivers, genetic drivers for each of them, we observed that the 3 of them were mostly dominated by the immune, vascular, and astrocytic components. These have a lot of implications. Again, data once again points to the immune system and the vascular system. And remember that there are multiple hypotheses associated with Alzheimer's dementia. One of the dominant usually has the prior life hypothesis or the amyloid cascade hypothesis. Where more and more work also points to the idea that the immune system and there is over-inflammation, there are vascular factors that also play a minor role in it. When we try to translate what we identified with postmortem data to in vivo data, we observed that the blood metabolomic and the regional MRI were the best predictors of the brain, of the pseudotype in brain. Essentially, we have here the results, and we have how well routinary blood tests, cell-free DNA, cytokines, monocyte RNA, proteomics, metabolomics, and regional MRI predict the pseudotype. Well, the pseudotype gives you an score reflecting how or the level of molecular alterations in terms of proximity to develop Alzheimer's disease dementia. And we observed that the best way to translate that to the clinic would be, at least in this scenario, with the blood metabolomic data. Then, when we try to see what specific data collected in vivo, we predict better to what subtype the person will belong. We observed that the metabolomics and regional MRI again will be the best predictors of AD dementia, but these predictions will work only when we compare with controls. In reality, we didn't find yet any in vivo data modality or most data modality collected in vivo that we predict with a high accuracy to what specific type of person will belong. Well, then we have some new work that in which we have improved the classifiers, but essentially we also know that only psychological trace information was able to predict to some extent relatively well to what molecular subtype person will belong. And that's also surprising because the psychological data is not apparently not done biological, but there is a lot of evidence that all psychological patterns relate to all molecular profiles in the brain. So we are doing more work to clarify that. But I think one of the main observations from here is that most of the predictions studies that we see out there might be misleading in the sense that when we focus only on one disease, or we see the whole disease as only one, all results will not really reflect what happens under the different variants of the disease. So there are many tools out there to predict who will develop Alzheimer's dementia or not. But that probably will work well for one subtype or the other subtype, or for the mix of all of them, but cannot capture, not necessarily will be able to capture the particularities of each subtype. And with me, I have a lot of implications too, in the sense that this person might receive, at the eyes of a clinician, the person is the same, but in reality, that's why the success of the current drugs to target Alzheimer's disease dementia is very low, because there's a lot of heterogeneity, and these drugs might only work for a small subset of individuals. Then, when we also did the exercise of trying to pass these pseudotypes and subtypes from the main database of cohort in which we discovered them to other external cohort, because that's the ultimate test, right? If these things are generalizable to other cohorts. And what we have here in the left is first how the pseudotype, well, we obtained data for other brains, and that was postmortem data too, and then we try to we train models on the Rosman cohort, and on the gene expression and proteomics information, and then we apply those weights to predict the pseudotype to the gene expression and proteomics information that we have collected in other brains. And then we observe, and that's what we have here, that the molecular pseudotype also predicts clinical dementia severity in the second cohort. And what we have in the right is that now we pass the pseudotype to blood data, but from the admin cohort, so again an external cohort. And this will be closer to a translational exercise. So when someone goes to the clinic, we try to do a similar test, like someone will go to the clinic, we will pass what we are obtaining in the postmortem cohort to that person that is going to the clinic and only have metabolomics information. With the idea of getting how far already the person will be to develop Alzheimer's dementia. And that's what we have here. We can see that the projected pseudotype, or the transfer pseudotype, is lower in the controls, like MCI means no cognitive impairment, MCI means the one in the center, mild cognitive impairment, and then on the right we have indeed. So we see that the transfer or projected pseudotype captures symptom severity in an external, in another external. It means that it's getting closer to get to a point which it could be used in the clinic. Although, of course, we still need to do a lot more tests with that. Now, I will briefly talk about some mechanistic models that we have been developing in the last years. So, about maybe 10 years ago, we developed in the lab a multifactorial causal model of brain reorganization and intervention, or we call it like that, in which essentially assume that from here, almost everything will be normally combined with molecular. This model assumes that the change that we observe in a given biology, in a given brain region, let's say, if there is an increase in amyloid deposition or an increase in vascular deregulation, an increase in brain atrophy in that region, that will be a direct function of all the change that happens that occur in that region, which will be contributing directly to the change that we are observing. So essentially, let's say, if we see increasing amyloid deposition in one brain region, that could be expressed as a function of the alterations at the vascular level, functional level, structural level, tau concentration of deposition, metabolic dysregulation of that brain region. Plus, incoming effects from other brain regions. We know that the regions communicate, but also we know that when there are specific alterations in one brain region, those genes propagate to other brain regions in multiple ways. We know that the typical ones, the Wallerian degeneration, we have a structural damage in one region, then there is demyelinization, there are other ways in which it propagates, then we start observing also atrophy in the connected brain regions too. The same happens with vascular deregulation. There is also a lot of evidence supporting that before the protein propagate from some regions to other. And for that, I will say later how we model that part. Plus, external inputs. Let's say if the person is taking drugs, if the person is doing physical exercise, and what kind of diet is following the person, and environmental factors. This general model can be mathematically expressed in terms of differential equations. This differential equation could be linear, could be nonlinear. We are working with both. What we have here is just a simple. It's just an example, but to say just to make it clear, what we have like ds here represents all the brain properties that we will be measuring. For example, it's an space vector in which we have like vascular values, amyloid deposition, tau deposition, atrophy values across the region, neuronal activity, etc. And then we model how these space vector change across time as a function of all the causal or direct interactions between the biological factors in which we are interested, plus the incoming effects from consumption. And then we have other equations in which we project this space and what happens in the brain to the cognitive level, right? So we always promote cognitive evaluations as a function of the multifactorial metrics that we have to characterize the brain. Essentially, this dynamic system analysis and control theory are used, as you know, in many engineering applications, from civil engineering to electric engineering in aerospace, so I don't need to enter too much detail here. But what we did was literally extending what the classical techniques that we learned on the engineering side to now apply them with multimodal neuroimaging. And in practice, what we do, we obtain different neuroimaging modalities for each participant. This could be amyloid, tau, glucose metabolism, all these ones are measured with PET. Also, we get blood flow, cerebral blood flow, measured with arterial streamlining, resting state or metric reflecting neuronal activity, and structural properties, which we measure with structural MRI. Then we just extract values for each brain region, and that's what we have here on the left. You see, there we have like almost around 80 brain regions, and then we just concatenate, the vascular values, the amyloid values, the neuronal activity values, and then we use differential equations. We have these across different campus for each individual. Then we use differential equations or model that is based on differential equations with some optimization technique to infer how all these factors are interacting, and also to estimate specific model parameters that are associated with the intra-brain propagation of their alterations. And that's how we finish with this causal or direct network that we have in the center for each person, in which essentially what we have are values, numerical values, reflecting how strongly the vascular system is impacting the amyloid system, and vice versa, and how atrophy is interacting with metabolism. And you get like a mechanistic explanation of how all these factors are interacting in Alzheimer's disease. We also get ways of reflecting how all these biological factors are impacting cognition. And so yeah, with all of this, you can have or you could recreate how the disease starts and how the different factors go from very small alterations in one particular brain region to minor alterations that also follow this intrabrain propagation for each individual. So what we have here, the brain is just how the alterations propagate from the initial disease epicenter, which we can estimate going back in time for each person. And on the right, we have the identified or reproduced intrabrain propagation and accumulation of tau, proteins, tau, amyloid, and gray matter atrophy. So you can see how the gray matter atrophy works each time for this participant that actually converted from mild cognitive impairment to Alzheimer's disease dementia, and we get this for each person. Yeah, this might be interesting or not, but still, I'm sure you have the question on how this can help the patient, right? So this is the initial model that we define. And now, if we are interested in, remember that at the end, you could also input information about what kind of treatment a person will be taking. Now, if you're interested in to make the invert question, we just need to invert the question, and we pass to the right side of the question what specific change we will need to do to cause a given. So we pass to the right side of the equation what specific change in the brain we would like to see, and to the left side of the equation what standard input we need to provide to the person to cause the change while maintaining all the other intents in the equation. So that's how typically control theory works, right? So by doing this, then we are able to simulate in the computer. Well, the person comes to the clinic or the person comes to the lab, we get all this imaging modality, or they might have the person might be characterized already. Then we apply the model, and we are able to simulate them. First, we estimate all the individual model parameters, and based on that, we estimate what specific biological signals we will need to be introduced to target the specific biological factors that we are considering at the individual level. So we have like an in silico simulator of how easy or difficult will be to conduce the person from a given clinical state to another clinical state, or to stop the neurodegeneration across time. And then this provides us something that we call the multifactorial intervention fingerprint. That is just like a virtual giving you information about which will be the ultimate intervention for each person. What we have here, for example, for patient number 1, is how effective will be to target the vascular system, the amyloid system, the functional system, the metabolic system, etc., or the tau, the position in the brain, to stop neurodegeneration in that person. And interestingly, we observed that for individuals that have the same clinical diagnosis, all these persons have the same clinical diagnosis, right? And this is a population of about 300 something participants in the Alzheimer's disease spectrum. We observed that for patient number 1, the vascular intervention and the metabolic intervention will be more effective than an anti-amyloid and anti-tau intervention. And there are some ways, to improve the vascular system and the metabolic system, like physical activity and ketogenic diet intervention. But for the patient number 2, who has the same diagnosis, an anti-amyloid and anti-tau intervention would be more efficient. And there are some specific drugs out there for that, which have been recently approved. For the patient number 3, nothing seems to be at the single target level, which seems to be efficient. And then we propose that for this individual, this could be for multiple reasons. This individual might have compromised different multiple biological systems at the same time, also might be more advanced in terms of molecular and biological alterations. However, we observe that if we try multiple or combinatorial intervention for this person could get better. So these are the kind of individuals that will benefit from a multifactorial intervention and not a single target intervention. We also extended this model to incorporate, until now we only use neuroimaging data, which, as you know, can be informative, but also is at most of the signals that we get only represent macroscopic alterations or macroscopic profiles. So we extended the model to also include neuroreceptors information. So we have whole brain maps of neuroreceptor density from a postmortem, a small postmortem cohort. And we use this map about 15 different neuroreceptors in the brain to, as a pure information to inform how the biological factors in the brain will be interacted. Remember that the previous model that I presented, in that model we considered, or we try to estimate how the vascular system impacts amyloid, how amyloid impacts the metabolism, etc. Now imagine that we just extended terms in the equation associated with those interactions to make them a function of the neuroreceptor densities in each brain region. And that allows them to estimate a specific biological parameters that reflect how GABA impacts or modulates or controls the interaction between the vascular system, amyloid, the atropine. So it gets a little more molecular, more informative at the molecular level. And in that sense, that allows us to analyze what specific neuroreceptors might be impacting at the individual level the clinical symptoms that we observe in Parkinson's and Alzheimer's disease. But also, we can infer what specific receptor and in what brain regions or brain areas are impacting or controlling the individual progression to a given disorder, to a given disease. Here, there are other things, but I think we could summarize all in this graph. And it was work done by American, who's a PhD student in the lab, now he's a postdoc. Essentially what we have here is this, something equivalent to a roadmap, which in the inner circle we have major macroscopic biological factors, for example, tau position, amyloid, cerebral flow, measure at the macroscopically at the regional level. And then next to it, we have a specific neuroreceptor system. And then out on the external circle, you have what specific neuroreceptors are controlling, and all the interaction between these of multiple pairs of neuroreceptors are controlling the change that we see in the immune cycle. So let's say that if you're working then on a given drug, we know we always, person that design drugs, they design them to target specific molecular pathways. And these molecular pathways are associated with genes, with neuroreceptors, etc. Let's say that if you develop a drug that targets the GABA system, then you go to the GABA system, you find GABA here, you project it, and you see how much this drug will be altering neuroactivity, or how much this drug will be impacting amyloid. So you can now, you have an information that will allow you to be more precise at the individual level in terms of the molecular system that you are targeting, and what, not only what specific molecular alterations it will cause in the person, but also what specific macroscopic alteration it will be causing, and the patterns of these macroscopic alterations. And you also know how that relates to the cognitive and the clinical level. So essentially you can go from the molecular level to the clinical level at the individual level. We also repeated this model, extended this model to include gene expression in the brain, and again we have whole mass of gene expression in brain, which we use as background information, and we finish with an equivalent map, in which now you have what specific genes. Say if you are interested for some reason in to target the cerebral flow, now what you need to do is to say you want to target cerebral flow maybe by the metabolic pathway, or first targeting the metabolic pathway, then you go to these specific genes, which seems to be the ones that are controlling the metabolic impact on cerebral flow. And you don't need to target, so you need to focus on drugs that target those genes, but that not only helps you to know what genes you need to target, it helps you to know which one you should not be targeting. Because essentially, the minor risks usually are the secondary effects of the drugs, right? So it's not only that if they don't work, it's that what else they could be causing that could be negative. And that's, you can use this kind of maps to decide or to even predict what will be the impact that a person will have when we use a given drug. Or even to design new drugs based on the information and the level of interaction that they trigger one that they are applied. In summary, this part with the therapeutic fingerprinting, we obtain different neuroimaging modalities for each participant, and then we apply these dynamic system equation models of framework to identify the current state of the person, or how the brain of that person is working in terms of all the biological interactions in the brain that we are allowed to see with the neuroimaging modalities. Once that we have the individual model parameters that we estimate with the longitudinal data, then we can apply functional theory to estimate what specific signal will need to be introduced to conduct this person from an undecided state to a decided state, which will be this decided state here in a multiple way, right? So the decided state could be just not getting worse, or could be getting better, or could be going back. And of course, there are limitations of going back. Sometimes we cannot really, there is limitation in terms of what we can do when we have a heavy atrophy in the brain. There are no ways to generate neurogenesis in the brain, but we can work with all those things to at least estimate what will be more beneficial at the individual level. And that's what we get here. So it's like what specific factors need to be targeted at the individual level, and then we can combine that information with the genomics or transcriptomic or proteomics information to know what specific molecular pathways then are underlying those changes that we need to apply at the individual level, or are underlying or determining why that person is progressing so fast or not to develop a given disease. Next time we talk also about all the work that we have been doing in the lab, and this was done by Yasser, who was a PhD student, now is a research associate in the lab. Which essentially and probably you are familiar with the neural mass model, but which allow you to simulate or to model neural activity in the specific brain regions, and then to see how they will they effectively propagate to the brain region. Essentially, like simulate neural activity in the brain depending on some specific biological factors like excitability, inhibitory activity, etc. So we're working on this model in which we assume that the neuronal activity at a given brain region will be a function of the excitability and inhibitory activity in that region, which at the same time will depend on the level of lymphoproteins in the region. So, and the interaction between these lymphoproteins. And once we have that, or the question for that, we just optimize the data in order to identify the ways of how much the lymphoproteins will be influencing at the individual level the neuronal activity that we measure from the neuron. So in this way, we finish having like specific ways of how amyloid, tau, all the interaction of amyloid and tau, and could be anything else like glucose metabolism, alterations, atrophy, etc. What specific ways they have on the excitability or the relationship of the balance between excitatory and inhibitory activity. And this is important because there is still a lot of questions about how these lymphoproteins impact neuronal activity to cause and neuronal activity, and how neuronal activity impact or cause the cognitive change that we see. So there are not still clear explanations to the whole process. So first we assumed that the model was able to reproduce classic neurophysiological patterns observed for Alzheimer's disease, but also importantly we assumed that the estimated model parameters of the association with amyloid and tau, the amyloid and tau neuronal activity was significantly associated with new biomarkers, amyloid and tau biomarkers measured in blood, which are used in the clinic right now. And that allows us to understand better in what specific areas and how strong is the impact that interaction have on neuronal activity, and what are the factors that contribute to it. But we are extending this model, recently extending this model to also include molecular factors, because although we hear a lot about Alzheimer's disease, and as I mentioned, there is a strong assumption that is caused by the lymphoproteins. We don't really know the molecular mechanism. We don't really understand yet the molecular mechanism that occur and impact neuronal activity. So almost all the knowledge that we have about why neuronal activity gets dysregulated and what gets affected is coming from animal models, which are simpler in some way because also they reproduce what we already know in humans. So they are created to reproduce what we know, with the illusion of somehow expectation that they will inform us about disease mechanisms that we don't know, but all could be a little simpler in that sense. So what we did was apply the same model, but now once that we obtained the model weights reflecting how amyloid, tau, the interaction impact neural activity, we try to understand what specific genes and proteins are associated with that impact. So in this way, we are linking the parameters that we are obtaining from the in vivo data and in vivo neuroimaging data under a specific biophysical model with gene expression and proteomic information that was collected when the person died. And within that, and this allows to then identify for the first time what specific gene proteins and molecular values are impacting or are determining or somehow are influencing the neuronal activity that or change that we see in brain that are caused by amyloid and tau. So now we are reaching what we observe at the macroscopic level with neuropathology with the molecular level. And I will not enter in details in terms of the molecular association, molecular molecular pathways, I just want to add that we also did the exercise of then training a specific, it was obtained with a given cohort, with a specific individual that have fMRI before dying, and then the molecular characterization after death. And then we trained it based on these molecular pathways and genes and proteins. We trained a model to see if they will predict in other individuals, independent individuals, how early or late we saw in terms of developing Alzheimer's dementia, and we obtained good results with those predictions. So essentially, supporting the idea that these genes and molecular pathways, genes and proteins and molecular pathways are having a major role on the development of the disease symptoms. So what we are doing next, we are trying to integrate all these models. We are always trying to extend them. We're also trying to validate them to get them closer to the clinical applications. We are also including considering lifestyle information, etc. I will talk about that at the end. But we are moving in multiple directions. One of them is like trying to incorporate more detailed molecular data. Although I was training on the neuroimaging community side, I know that at the end we really want to say that we need to reach all the most data layers possible. So there is no single data model that will give us all the answers that we need to extend and to validate the diseases. So a work that we did recently is like based on RNA velocity fields. So I talked before about using single cell RNA data. Essentially what we get with this is just like a snapshot of the gene activity in this cell. And that were, because this data used to be collected from a sample, and that sample is usually dead or not dead, but this sample needs to be somehow, it's not a living sample, right? So we get out this snapshot or like a picture of how the genes were activated at that time, but we usually cannot infer from there where they were going in terms of activity or what else was going to happen. So RNA velocity is a new concept introduced like around 2018 that then allows to, based on the relationship between the new, let's say the ratio of gene creation and the gene consumption or gene activity degradation. Based on that, it allows you to estimate the velocity of the change in that gene. So what we finish is having not only the activity level, but also how fast that gene was changing and in what direction. Right? If it was negative, it was positive, it was increasing or going down. So in a way that you could, if you have, let's say, only one time point, you could infer what will happen a little after that. And you also could represent all these, the RNA velocity in these two-dimensional fields, in which you can see, or you can represent the information flows across the different cell types. So if the gene expression will give you just what is in the left, then when you get the flows, you have an idea then of the causal mechanism of how the disease is propagated in one direction or the other, how the process are going from one type of cell type to another type of cell type, and what specific genes and molecular powers are involved in that transition. And we estimated the RNA velocity field for normal individuals with no Alzheimer's dementia individuals across different transition states, and we can see there are a lot of change in the flow of information, of the way that the cells interact, depending on what stage is this person. I'm trying to consider that information incorporated in more models too. In addition, we have inferred cell types proportions in the brain. So now we have detailed maps of what are the densities of microglia in this brain area, or this specific ball cell, neurons, how much, and what is the proportion of neurons in that ball cell. And we observed that, well, first, this is very important because at the end, all what happens in the brain depends on the intrinsic molecular characteristics and structural characteristics of each specific region, right? And what we observed is that in those regions, there is a strong association between the patterns of atrophy that we develop for different neurodegenerative diseases and the patterns of cell densities on those regions. So the regions, let's say those regions that there is an imbalance between microglia and neurons, they tend to develop more atrophy for some specific diseases. And this already gives you an idea of what specific biological mechanisms are determining the diseases that we develop. So maybe, say, if our brain is configured in these specific ways in which, say, for one person, in which for some reason there's a little imbalance between the relationship between microglia, neurons, astrocytes, in a specific brain region, then we will have a higher probability to develop one specific disease that is associated with that one. And then if we have the characteristic, different characteristics in other direction, then we will develop the other disease that is more associated with those other characteristics. So it's all determined by how our brain, grow and how these cell types are concentrated in across all the brain regions that we have. And among other factors, I don't want to just say that these are the only things that matter. We are also extending this to consider cell types interaction. So it's not only about how they are distributed cells, or the different cells are distributed in the brain, it's also about how they interact. So it's interaction that could give you, or analyzing interaction among brains, but could give you a deep mechanistic understanding of how the disease happens. And we are using this now to understand better Alzheimer's disease, or cell-cell interactions in Alzheimer's disease. And just to finish, implementing all these computational models, as you know, takes a lot of time and try and error. So we have also developed OpenAnswers, a user-friendly software, a user-friendly software in the lab that allows to go from the neuroimaging data, the molecular data, treatment information, to apply the different models that I presented, and get also some visualization of the results. And there are other, we also share all the codes that we develop in the lab. There are other studies submitted with new methods proposed that we are also sharing with our GitHub. I would like to some ongoing work. We also working in the blood and CSF stratification of Parkinson's disease. We are using the single-cell RNA derived data to estimate, to understand better the cell-cell interaction, but now the single-cell load. And we, as you know, you might be familiarized with the brain connectome studies, and we worked very early on that too, like some of the first generation for the structural brain connectome. And there are others groups that are working on functional connectome, and I believe they are working with functional connectivity too, a lot from EEG and other methods. But now we are trying to also infer molecular connectomes. So connectivity, or we have specific ways to estimate how the different cell types and ligands and receptors associated with these cell types communicate within specific brain regions and across brain regions. So in a way that we are trying to construct, we already have some version of it, but still haven't published it. We are constructing these cellular level of brain connectomes that will allow us to understand multiple biological processes, but also how the microscopic connectivity that we normally estimate with the MRI and other techniques can be decomposed into cellular connectivity modes. And in addition, what specific cellular interactions are causing the progression of Alzheimer's disease, dementia, Parkinson's, prevalent neurodegenerative condition. And finally, we are adding lifestyle, environmental information, social interaction information, like all of this information that could be, that is associated with all the things that happens in our life that could be represent modifiable risk factors, with the idea of identify what specific interventions we need to also plan them to make them remain. So that I'd like to say, right?
If you didn't have an idea what personalized medicine was, this is what it is.
So it's really, really cool. Can you talk a bit about the challenges when you're building these models, multi-layer models, of how different these variables are too, like from measures of cognitive function to the very complex spatial mapping from the clinical imaging to single cell continuous maybe transcriptomic data to categorical environmental factors. How do you put all of that together?
Yeah, so they are very different, and it depends. Like if we are planning to use, so we have these two main analyze them, their patches, effects, but essentially just using advanced technology, they are integrated, the practice and dimensionality techniques, give you an idea which ones are having more weight, and you always can go back. It's a little bit more challenging when you work with the mechanistic models because they are based on assumptions about how things usually interact on this. So we do kind of a combination. Let's say we only have one modality, we assume it's by physical models or models in which everything interacting with everything, and without having to assume, because all of them are information in the same brain areas, and that's relatively simple, that's what we can say. But if we are analyzing, let's say, social environmental factors, then we need to combine this mechanistic model with more like empirical model. We have like parallel equations in which we just estimate ways to go from one direction to other, but without assuming given any brain path. So we have general parameters, the optimized parameters that give you an idea of the mechanistic sense there.
So, thank you. So actually, you said that to predict the Alzheimer's disease or dementia, blood, MRI, or psychological information. So I'm curious about what kind of blood-related information you are used to predict Alzheimer's disease or the dementia.
So there is a lot of work now in the last year, in the last decade. We see more and more work. Actually, there are some markers already approved for clinical application, like predicting AD progression based on some specific proteins. We have these proteins are getting very popular. If you go to any major AD conference, before we were only maybe it was only talking about MRI. Now it's almost only talking about the blood markers. It's on MRI too, but what predominates are these blood markers. Based on my experience, I observing that proteomics is very valuable for it, for making this prediction, but also metabolomics seems to be strong predictor too. I'm not sure which one will be more best, but I'm betting a little more on metabolomics.
And you said that MRI related information, that means you are trying to say that fMRI related information, that means blood oxygen level dependent information you are using for predicting the Alzheimer's disease or the dementia?
Yeah, so there are different markers too that you can get in that sense. You can get like reactivity or measure reactivity, fMRI, maybe metrics. And we try to make them compete to see which one will be the optimum at the end. Yeah, we don't have a final answer to that.
And the last question.
Yeah.
And in your multifocal mechanistic model, you use different elements like vascular element, amyloid element, functional element. So I'm curious how you model the vascular system to see the impact of this vascular element of this Alzheimer's disease and for getting the corresponding therapeutic strategy.
Yeah,
so
so
how you model this vascular system?
So we start the cerebral flow from the aggressive labyrinth, that's one way, and then we just have this. Assume that there are interactions between the vascular system of the signal that we're estimating at the regions and amyloid, tau, like the other parameters. So we use the individual data to optimize those more parameters and then give us some understanding on how the vascular system is impacting the others. But also we train models to see how the vascular system in combination with the other factors are might be impacting cognition, clinical progression.
Okay, thank you again. We may have the lunch with some students if you're interested. The seminar